Hello, welcome back to my Colloquium 2 video. This is going to cover class 5, which is the second class of Colloquium 2. And in this class, we're going to look through the brain development, especially when it's more zoomed in, like it is in a microscopy slide. What signs we're looking out for in terms of brain development, which structures in the brain are the most important structures, as well as some other head structure derivatives that we need to know. Other than that, we need to learn about the abdominal organs, such as the small intestine development, the liver, the pancreas, as well as some intestinal loops. The lung in pseudoglandular stage hopefully is something that we can all recognize, but today we're going to go through the structures within the lung. As well as the developing neural tube, which is a primordial version of the spinal cord. Let's start with the most complicated slide of this class, and maybe even the entire colloquium, which is the developing human brain. Now, the developing brain is complicated because we're not going to have this clear of an image in the colloquium. I hope that is kind of understandable. These are images that show you as far as the microscope can actually zoom out. So if you can't really see the signs of where you're located, it's going to be difficult to actually understand that this is the human brain, right? So that's why I'm here, hopefully to try to make you understand the signs that are important to look out for in terms of knowing where you're at, and then also the most important structures of the human brain. So let's go back to this. So a slide like this, according to the professors at least, is not something that might appear in the colloquium because we don't have the characteristic signs here that can help us identify where we're located at under the microscope. For example, one of the most important signs are the tongue and the eye in order to understand that we're in the head area, especially the eye when it comes to the brain development. Instead, we're going to have a slide that looks more like this. So from afar, it doesn't look that characteristically skull-like like this one does, right? This one has more of a head-like shape. However, when zoomed in, we're going to be able to see the eye development. Hopefully you'll recognize that this is an eye. Even though we haven't really covered that, we're going to cover it in the next video. But as soon as you see the eye, you will immediately be able to understand that we're in the head area or head region, and therefore the possibility or the chance that we're going to have or look at the brain is very, very high. So usually professors will show you the slide first, like in its entirety, zoomed in, and then they'll show you especially those important markers that will help you recognize where you are, and then they'll get into the actual slides or the structures that matter, for example, the medulla oblongata. Now, in my opinion, the structures that I found the professors talked about the most, i.e. were the most important, were these following structures. It was the nucleus caudatus, and then with the nucleus caudatus came also the migrating neuroblast or the neuroblast migration. We can't really see it here, but we'll explain about it later. The neocortex, the plexus choroideus, which we could see in a lot of different areas of the brain, as well as the medulla oblongata, pons, and lamina quadrigemina. Those are just the parts of the brain that I felt were the most important, but again, ask your professors. For example, the developing cerebellum is still an important structure for you to learn about. It's just more difficult to show it in a slide and be absolutely certain that this can't be mistaken with another structure. The thalamus is a structure that I didn't find the professors talked about very much. But again, they might be watching this very video and just like rearranging the structure of the colloquium. So talk to them, ask them, and hopefully it'll make more sense then. Another thing that might be important for the colloquium that I didn't find that vital, but I would like to bring it to attention anyways, are the ventricles. So for this colloquium, at least for the practical part, you need to know about the third as well as the fourth ventricle. Now the third ventricle is just above the eye towards the front. It's also where you can find the nucleus caudatus, while the fourth ventricle is where you can find the medulla oblongata. Again, is this something important? Are they going to ask you about which ventricle is where for the practical part of the colloquium? I doubt it, but I would like to bring it to attention because that's what our professors did. Let's start by talking about the medulla oblongata, the pons, and the lamina quadrigemina. These are one of the most identifiable structures of the brain, and it's also one of the 3 structures or landmarks that I would like you guys to know about in regards to the general brain area. Now, if we look at the no annotation slides, I've tried to draw this little guy here that will represent as part of the brain. So first of all, the head here with the little nose, you can see here, I've tried to draw it. This would be the thalamus. A little bit of a less important structure, but we'll get to it soon. Then the general thorax or body area. Then we have the little behind here that would represent the lamina quadrigemina with the kneecap going down towards the bottom legs here with the little feet. If you can see this little guy, hopefully you can immediately recognize that the pons, this area looks like a kneecap, while the medulla oblongata looks like the bottom of some feet or below the knees, and the lamina quadrigemina looks like the thigh or behind area of a person sitting down. This is what I would like you guys to imagine during the colloquium. Now, if we look at the demo lab slide, this is something that appears almost always. So here, we don't see the remaining parts of the guy, but we can definitely see the thigh here, or the bottom area, which is the lamina quadrigemina, the kneecap here, which is the pons, and below the knees, the feet area, which is the medulla oblongata. If we look at other images, we can definitely see it more clearly, but this is what it would look like under the microscope, because again, we're first going to see the eye, which would be here, and then from the eye, we're going to go towards the bottom left area, towards the fourth ventricle of the brain to see the pons. This is how zoomed in it will be, the pons. Hopefully that makes sense, almost looking like a kneecap, the lamina quadrigemina area, as well as the medulla oblongata. Like I said, this is how zoomed in it will be. If we look back at the images, there we go. This is another one. So here we can't see the eye super perfectly. Well, we're actually missing the lens, but hopefully that still makes sense that this would be the eye area. Then towards the bottom left of the eye, like I said, it could be upside down. We'll get to it soon. We have these 3 structures. Now, like I said, this is the little guy's head. And then we have the bum area, which is the lamina quadrigemina, the kneecap, which is the pons, as well as the bottom legs, which are the medulla oblongata, or is the medulla oblongata. We can look at it from another one. Here we go. Here's another one. This one is actually the one we looked at the demo lab, only in a more e-studies context. So again, the eye right there. Then we have the inner ear development. We'll get to it soon. But this kneecap is the pons. This behind area is the lamina quadrigemina, and then the feet and legs below the knees is the medulla oblongata, with the face here being the thalamus. I believe you have to write in developing thalamus, developing lamina quadrigemina, developing pons, developing medulla oblongata, which I unfortunately didn't do for the annotations. So please keep in mind that if we have these structures as full-grown adults, and we're looking at an embryo slide, which this is, obviously, then always write in developing before the actual structure you're given. Yeah, that should be it for the medulla oblongata. Now, if we do look at this from an upside down angle, for example, something like this. In one of my lectures, the slide was upside down, so the chances of it being upside down for your practical colloq might also be very high. So here we see the eye development, and now usually we would go towards the bottom left area, right? But here we have only empty space. Also the placement of the lens being towards the left, or the general eye being towards the left instead of the usual right that we see here. Or this would be better. Here the eye is towards the right, and then there's empty space towards the right side of the eye. Here there's empty space towards the left side of the eye. That means that everything is upside down. And now instead of going towards the bottom left, this area, which is also, like I said, upside down. So here we have the pons in this case. Then the feet below is the medulla oblongata, and then the behind area is the lamina quadrigemina. I think that made sense. Now let's continue on with the next structure. Now, the second structure I'd like to talk about is the nucleus caudatus, and it appears like this. It's very close to the general eye area, and if we look at it with no annotations, it appears almost like the head of a mushroom, almost like a papilla of the tongue, right? Protruding out like this. And usually the nucleus caudatus has additional structures that I would like to call migrating neuroblast. The best way I could describe this, here we go, this would be the nucleus caudatus, and then we have the migrating neuroblast right there. The best way I could describe this general structure is if you imagine like a jellyfish. So a jellyfish has like a little head area, and then it has these tentacles, little wisps, and that is the best way I would imagine the nucleus caudatus. So here again we have the general, it's lagging a lot, sorry, the general head area of the little jellyfish, or the nucleus caudatus in itself, and then all of these little wisps that we know to be migrating neuroblasts, looking like a connection of veins, like so. We have it sort of up here as well. This would be the nucleus caudatus, and then we have some strings here that we can safely say are migrating neuroblasts. The nucleus caudatus has an additional area, or an adjacent area, called the capsula interna. Now this is a structure that not a lot of professors think are as important, but Professor Vaivads did mention it, so I do include it just in case. Now to look at the demo lab images of the nucleus caudatus, we have it here. So for example, they would show you the eye as a landmark or a signifier that you're in the head area. Then they go towards the left, and closest to the eye we see the nucleus caudatus. Now that is the top of the nucleus caudatus, and then we have the small jellyfish legs, which are the migrating neuroblasts, just like so. I think it's a generally easy structure to recognize, but it's also one of the 3 most easiest landmark structures for you to use as localization. So again, nucleus caudatus is within the 3rd ventricle, like so, or closest to the 3rd ventricle, and the lamina quadrigemina, pons, and medulla oblongata area is towards the 4th ventricle area. So that is how you can localize all of the structures. So like I said, these 3, in addition to the eye, are the most easiest and easily recognizable structures in terms of localization. I think that is it. Let's talk about what it would, that's another document entirely. Let's talk about what it would look like if this slide were to be upside down, for example. So let's zoom up, and we see the slide being to the side, maybe something like this, right? Now here we can still see that the eye is very much visible, and the outside of the eye, or the empty space, is towards the top of the eye. So this has been sort of moved at a 90-degree angle. Now if we go towards the bottom, closest to the eye, we have this area. Now here it's a little bit harder to recognize. Sometimes some people will mistake this area, which is the thalamus, with the nucleus caudatus. The thalamus very much looks like the nucleus caudatus from some angles, but the nucleus caudatus is a lot smaller in size, and it's also closest to the eye. This one's a little bit further away from the eye. In addition to that, the thalamus might seem like it has those tentacles, but it's more prominent with the nucleus caudatus. Now, if we do this, and we imagine, it's lagging again, there we go. There's a lens here, and this is the general eye area. Now, if we go towards the bottom left-ish area, we can see the nucleus caudatus more prominently here with the migrating neuroblast. We can see the thalamus as well very prominently, but the best way I can describe it is that the nucleus caudatus has a clear, dark edge or corner compared to the thalamus that's very light in comparison. So here you can see it again, the clear dark area compared to the thalamus, and then we have it here as well. So if it were to be, let's say it's upside down this time, right? So it's upside down, we have the, let's let it load. Okay, there we go. We have the eye there, and then towards the bottom like that, we have the nucleus caudatus that is very much darker at the top compared to the general thalamus area. We can also see very clearly the migrating neuroblast here. Another signifier that we're looking at the nucleus caudatus and not the thalamus is this sort of tail that sort of protrudes out. Let's bring all of these back to their original forms. There we go. Now, if we look at this general area again, we'll look at the nucleus caudatus, we can see that it has this little tail protruding out of it. So the nucleus caudatus is here, and then it has this little elephant-like nose that protrudes out like so. So that is another way to identify that we're looking at the nucleus caudatus and not the thalamus, which is areas that we generally mistake for the other. So there. However, I find it that like 9 out of 10 times, the professors generally talk about the nucleus caudatus, not the thalamus. So making the mistake, if it comes to it and you're deliberating between the nucleus caudatus or the thalamus, I would say write the nucleus caudatus if you're unsure. If you're sure, write what you're sure about. But if you're unsure, probably write the nucleus caudatus, because that might be the area that they are actually interested in. The next structure I'd like to talk about is the plexus choroideus. Now, the plexus choroideus appears as almost this wishy, like almost fabric-like, close to loose connective irregular tissue that sort of wisps around the main material of the brain. So you can see it here as well. Again, very wishy-washy. It'll be more clear once we zoom in and show you the actual structures. Without annotations, you can see it here. But let's move on to the next. Here we can see the plexus choroideus more in detail. Like I said, very wishy-washy, very wiggly. It's the wiggliest structure in the brain. So if they ask you about that, I would just say the plexus choroideus is a easy guess to make. We can see it here as well, very wiggly, like I said. In another structure, here it is again, and then there it is more in more of a mass, I would say. Again, very, very, very wiggly of a structure. If we look at the demo lab slides of it instead, here we go. Do we have any here? Here we do have, this is part of the brain, and we can see here that we have the plexus choroideus right there. Again, very wiggly of a structure. I'm repeating myself with the adjective I'm using to describe it, but I think it makes most sense like that. Here we have it again, and then there, that would be the plexus choroideus, I would say. But more detailed, here we have the pons. This is the medulla oblongata, lamina quadrigemina, and then down here we have the plexus choroideus. close to the medulla oblongata or close to the nucleus caudatus. She can't really, here we go, nucleus caudatus. We also have the plexus choroideus, but it could appear in other places of the brain as well. I'm just going to say it here that if you see a wiggly kind of structure in you know that you're within the brain, general brain area, and it's next to the nucleus caudatus, next to the medulla oblongata, but could also be next to other adjacent brain structures like it is here. You will just, it's an easy guess to assume that it is the plexus choroideus. Following the plexus choroideus, I would like to talk about the neocortex. Now, the neocortex, a lot of people make the mistake of mixing this structure up with this structure, the neural retina of the developing eye. This is a structure we're going to cover in class 6, but to sort of mention those two and compare them, the neural retina is obviously in the eye and the neocortex is within the brain and the skull. Another important difference is the size, but of course you're not going to be able to compare the two, but the most important thing is that the neocortex is part of the brain and the neural retina is part of the eye. And the neocortex, it continues on. It's has this snake-like formation. It almost looks like a neural tube. It's not the cerebral cortex, which we can see here as a little bit larger than the neocortex, but thankfully we don't have to know about the cerebral cortex. So if anything looks like the cerebral cortex and it's within the brain, then it's most probably not the neocortex. Let's look at some other places that we can find the neocortex. Here, for example, this would be the neocortex general area. Here as well, the neocortex. Thankfully, we don't, even if we make the mistake of mixing it up with the actual cerebral cortex, it doesn't matter because the neocortex is the only thing that appears in the structure list, though double check that with your professors, please. Other than the neocortex, which I think is a fairly simple structure to notice because again it slithers, let us actually look at the demo lab slides before we continue. Like I said, we have this darker outside and the lighter inside. I would say maybe this could be the neocortex. This looks a little bit too large to be the neocortex, but it could just as well be. Again, it all depends on the context. But yeah, definitely if they ask you about this general area, then I would say it's the neocortex. Let's look at other areas if we can find it here. Perfect, the neocortex next to the plexus choroideus. So in that case, I'm guessing this could be the nucleus caudatus, unless I'm wrong. Let's carry on to the next slide, and I would like us to talk about the cerebellum. Now, the cerebellum is a very difficult part of the brain to identify. It's easier here because you can immediately just assume that this obviously is the cerebellum. However, going down, because it's cut in a way, the face is not cut evenly. Like if we have a little guy here, usually it's cut in a very even and symmetrical sagittal section, but in this case it's cut in order to gain part of the eye. So for example, it's cut like this, so we have part of the eye for us to be shown, and we have a little part of the brain, but not a symmetrical part. Okay, and because of that, it's hard to see where the developing cerebellum really is. So some professors say that it is this area that is the developing cerebellum. Some say this area. Depends on the professor. Ask your professor. Obviously, the developing cerebellum can't possibly be this large. Double check that with your professors. I personally didn't study a lot in regards to the practical part of where the cerebellum is, unless it was very, very vivid. For example, in this case, this is a very vivid part of the cerebellum. It looks very cerebellum-like. Not only that, I think we have a separate, there we go, separate little image from e-studies that really show you the cerebellum part like so. And but not all slides will show you the cerebellum that detailed. So that's why a lot of people, might assume that this is the cerebellum or this instead of this. It again all depends on the professors. A lot of professors say different things. Double check it with your professor, make sure that you know where the cerebellum is, and make sure to ask if that is a prominent structure to remember for the colloquium. Now that we covered parts of the brain, let's talk about parts of the skull. Now we have a desmocranium towards the top of the head, and we have a chondrocranium towards the back and bottom of the skull. The desmocranium, in my opinion, looks a lot more frail and fragile. It looks darker, it looks very, very thin, and like I said, it's above the eye and covers the part of the head like so, like a forehead structure. While the chondrocranium, we can pretty much see almost at the bottom of the face towards the mouth area even, and it's a lot thicker. It appears very much like a hyaline cartilage, while this appears very darker and almost like it's about to shatter and fragment. Let's look at other parts. Here we have the desmocranium, like I said, very dark in appearance and looks like it's about to shatter. It's very, very thin. While here, the chondrocranium, we can see again, very hyaline cartilage-like in shape and appearance. Can we see other parts of it? No, we can't. This is just the part that we can see. If we look at another slide, here we have the chondrocranium. Again, we can see all of the different chondrocytes, while the desmocranium appears very dark and again above the eye. However, we do see these structures in other types of slides as well, not only that type of slide. Let's look at the head from sagittal section. I believe we can see it here. Yes. So this is an animal. I think it's a dog. Yep. In sagittal section, we're going to get to this in class 6. But to make it more apparent for the different types of skull sections, here we have the desmocranium. In this case, it's blue because we're using a specific dye that dyes this specific material blue. We can also see the intramembranous ossification in blue. You can kind of compare the intramembranous ossification shape and like appearance with the desmocranium, only the desmocranium has one line like so. While the chondrocranium, like I said, appears very hyaline cartilage-like and is a lot thicker. It can also be towards the bottom of the head. Maybe this, unless it would be more clearer if it was more zoomed in. Let's look at other areas of the head. Do we have them here? Won't say that. Here we go. We have the desmocranium here, again, blue in color. And then the chondrocranium here, back of the head towards the bottom. Here we can definitely see it. This entire thing would actually be the chondrocranium because we can see all the individual chondrocytes there. While the desmocranium is more of a forehead type of skull near the eye, the chondrocranium is thicker, more hyaline cartilage-like towards the back and bottom part of the skull. Here we have it again, desmocranium, bluer and thinner, more fragile, and then this would be the chondrocranium. I just didn't annotate it. As for examples of what these two parts of the skull look like under the microscope, the most important thing I will mention that the eye is not only a great landmark of where you're at or that you're gonna see sections of the brain, it's also a good indicator of where you're or what type of skull fragment or skull part that you're going to look at. For example, if we go towards the top of the eye, closer to the empty space, like you see here, so this would be just like slightly towards the top area, that is where you'll find the desmocranium. All of these smaller parts are the desmocranium. Instead, if you go towards the bottom left, close to the general medulla oblongata, lamina, quadrigemina, and pons area, that's where you're going to find the chondrocranium, just further down here. So for example, this would be the medulla oblongata, pons, and lamina quadrigemina. Here is the eye, so further down like so, that's where you're going to find the chondrocranium, and it looks and appears very hyaline-like in cartilage as well. Before we get into the developing inner ear, I would like to talk about an additional bone-like structure that is close to the eye, without really delving into the development of eye, because again that is from a structure in class 6. The structure I want to talk about is the vacuolized orbital bone, aka a type of hyaline cartilage, right? And we can find this right beside the eye, towards the area close to the brain development. So all of this is known as the hyaline cartilage, but you can also just call it the vacuolized orbital bone. Maybe your professor wants you to mention this name instead of this name, but our professor thought that both would gain you full points. We can see the bone in other areas. This would be the eye, it's just missing its lens. And then we can see the hyaline cartilage-like structure right there, the vacuolized orbital bone. Right here. And then here we can see it right above there. That would be the vacuolized orbital bone. Some would also say maybe this. Again, depends on the professor. If you see any hyaline cartilage-like bones towards directly towards the back of the eye, it's 9 out of 10 times more likely to be the vacuolized orbital bone, aka the hyaline cartilage, than anything else. Finally, the inner ear development. We are going to cover most of this in class 6. I am going to say, however, that this is one of those structures that a lot of professors disagree about, not to their detriment per se, but that the inner ear is such a delicate organ. It's very hard to distinguish where the ampulla lies, what the cochlea are, what the semicircular canals are. So all of those different individual structures we're going to cover. But to give you a general tip, the inner ear obviously is towards the ear portion of the face. It's towards the bottom left of the eye, and it contains or it's covered by this area called the cartilaginous capsule. So if you see something that looks oval in shape, it looks like bone formation and it has some holes in it, then it's probably the inner ear development. In addition to that, all of these holes, if you have to guess, just call them developing cochlea, if you have to guess. It's hard to distinguish between semicircular canals, ampulla, cochlea. I think there's another one that I'm forgetting, but all of those structures are very similar, and there are barely any slides that have clear differentiation between them. So that's why if you see this type of structure and it's covered with this bone-like formation called the cartilaginous capsule, the holes are probably just developing cochlea. But we're going to talk about the developing inner ear later. We finally get to graduate to another slide, and here I would like to put more attention to the developing spinal cord as well as the developing neural tube. Now, when do you use neural tube and when do you use developing spinal cord? The neural tube is a primordial version of the spinal cord, and I think it's up to the 4th week of embryonal development that it still persists, and then it becomes the spinal cord. So in this case, I would say we're definitely past 4 weeks of gestation, so probably this is more of a spinal cord. However, when it's isolated, like this is the longitudinal version of that structure, then the professor adds the name neural tube. So ask your professors. Thankfully, though, I noticed that instead of asking about the whole general structure, they ask you about the different layers of the neural tube, which is exactly the same or are exactly the same layers as the developing spinal cord as well. So that is a great compensation, I guess. But going back, let's talk about where we can find the spinal cord / neural tube. In this case, we're seeing a more sagittal section towards the face. This is actually the inner ear development. So we'd probably have the eye here, the brain being here. That's a great image of the brain. And then here we have the start of the developing vertebral column. And the vertebral column obviously envelops the entire spinal cord. Now this is a transverse section of the spinal cord, so we see the lighter area that will develop into white matter later, known as the marginal zone, the a little bit darker area that is larger than most other layers, known as the mantle zone, and then the much darker area in the middle known as the ventricular zone, and that opens up to the canalis centralis. Only here it's cut in a weird way, so we get like a little bit of canalis centralis and a lot of the ventricular zone. Surrounding the spinal cord, we have this tissue called the pia mater, in this case the developing pia mater, and obviously, like I said, it is the spinal cord, so we will find it within or enveloped by the developing vertebral column. I'll show you what that looks like in a second. And as for a structure that we recognize from the previous colloquium, we can also find the developing spinal ganglion. And some professors point out that it is important to know different sections of the spinal ganglion. I say not so much, but they might change it in the future. But looking at other ways, we can see this developing spinal cord. So here I've shown you 3 different slides. Obviously, they're not going to be this zoomed out, but first let's get a general idea of where we can find the spinal cord. So the spinal cord we find close to the vertebral column. I hope that made sense. So if you look at a more traditional sagittal section of the embryo, where do we find the vertebral column? Here, right? All of this is the vertebral column. These are the ribs, although they're a little bit weirdly cut, but here we can definitely see that vertebral column with the corpus vertebrum, probably this, and then the processes of the vertebral column. And then in the center, we see these openings, these things that poke out. These are most probably spinal ganglion, but more deeper here, we can see here the vertebral column has opened up to the developing spinal cord. And here we can find the different layers. So we have the primordial white matter, known as the marginal zone, because it's the marginal area, right, within the margins. Then we have the much more abundant mantle zone, and then this very, very dark area that surrounds the opening of the canalis centralis, known as the ventricular zone. Here's another sagittal section of an embryo, and here you can definitely see all of these vertebral columns opening themselves up for this spinal cord that they're hiding within. And here you can, I would say mainly you can see the marginal zone and the mantle zone. You definitely should be able to see this, which might be the pia mater, which surrounds the spinal cord. Do we see it here? Yeah, this area, or this in general, would be the pia mater. Here we actually have the transverse section of the embryo. And what do I mean by that? Well, if you imagine the cut being from the belly button to the back, sort of almost cut like a cucumber, that is what you see here. So here you see a pretty large organ, and this is the liver, and this would be the lungs. I hope the texture is recognizable to you. We have the esophagus as well, that is like the tube that it sits between the two lungs, and we're going to get to some lung structures later, as well as the liver structures later. But here you can see this thing that separates both of them, and I hope it is recognizable as the diaphragm. Separates the abdominal area from the thoracical area, in this case. Okay, now what I would instead like you guys to look at, to get back on topic, is this little thing. And here we can see the vertebral column cut in a transverse section. We have the corpus vertebrum and the different processus vertebrae right here and here. And then in the center, it envelops the spinal cord. And the spinal cord has a lighter area known as the marginal zone, the darker area known as the mantle zone, and then here it's hard to see, but it is an opening neural canal with a dark rim known as the ventricular zone. And then we have the surrounding tissue known as the pia mater. So again, the spinal cord might appear longitudinal like it does here, or it might appear transverse like it does here. It all depends on the slide. Here is another transverse section of the spinal cord. We have the corpus vertebra, we have the arcus vertebra or the processus. And then here in the middle we have the spinal cord, and you can see here that we have an opening known as the canalis centralis, the ventricular zone, which is a lot darker surrounding it, the mantle zone, which is the not as light as the marginal zone but lighter than the ventricular zone, and it has more of a bigger mass than the rest, and then the marginal zone, which is this wider area. And then of course the pia mater. Surrounding the developing vertebral column, we have these ganglia known as the developing spinal ganglion, which is definitely something that might still appear in your colloquium. Here we can also see the developing pia mater, almost looking like the Plexus Choroideus that we mentioned before. Here we have the developing truncus sympathicus, which Professor Vaivads thinks is very important for the colloquium, so please keep that in mind. But I would say spinal ganglion is a little bit more important, other than the structures within the spinal cord. You don't have to know about the corpus vertebra or the arcus or anything like that. What's important is for you to remember that this is a vertebral column. Okay, here we go. Here we can see in longitudinal cut the neural tube, aka the spinal cord, but in this case it's a lot younger, so this would be the neural tube. And here we can see the opening, the canalis centralis. Now it's again in longitudinal cut, it's not cut in a transverse section like this. And then we have the marginal zone, which is this wider area, the mantle zone, which is a little bit darker and larger, and then the ventricular zone, which almost appears black if you zoom out more. And then we have below the marginal zone, we have the developing pia mater as well. Okay, and here we have another version of that, just in a little bit worse of a quality. You might expect the spinal cord, the developing spinal cord and neural tube to be separated like this, but in reality, usually when the professors show you these structures, they will probably be in more related structures. Now here it is separated, like you can see here, but actually it comes in probably like a sagittal cut of an embryo. We can see it in another image as well. Like I said, it will probably appear in a more sagittal section of an embryo. So if you're finding yourself in the sagittal section and expecting that we're gonna talk about organs like the liver, lung, etc., you might have to double check that, because it might just be that we're more focused about the neural tube and spinal cord instead, as well as the spinal ganglia in relation to it. Before we get into the organs, we have to figure out where the organs are actually placed, and usually they're seen in the embryo and sagittal cut. They're also seen in the transverse cut of the embryo, but it's usually easier to view which organs are in the transverse cut rather than the sagittal cut. The most important way to figure out the embryo and sagittal cut is to find the abdominal area in comparison with the thoracic area and vice versa. For example, the thoracic area is going to be above the diaphragm. It's also going to host organs such as the lung in pseudoglandular stage, the developing heart, as well as other structures, but we'll get to that in a second. We're also seeing the developing rib here. So as soon as the developing rib ends, that's where the thoracic area ends as well. And instead, we start with the abdominal area. Now, the abdominal area hosts the organs that we know from the previous class, which is the metanephros, the developing adrenal gland, and then some structures from colloquium 1 as well. But most prominently, we can see the liver in the abdominal area. So again, if it's going to be very zoomed in a microscope, and if you see the ribs, that's a signifier of you being in the thoracic area. Not to mix the ribs up with the developing vertebral column. Like you see here, these also appear like ribs, right? Circular, just like these do. However, the ribs and circular. There are no additional structures for the ribs. They're all just circular in one line. Sometimes they can be a little bit more oblong, but that's pretty much it. Like, they don't continue there. There's not additional bone development occurring here. While for the vertebral column, you can see here, even though we have the circles here, we have additional development here as well. So that's how we can easily distinguish the developing vertebral column from the developing rib, because the vertebral column we can find both in the thoracic area as well as the abdominal area. And we're just interested in the developing rib so that we can identify where in the body we're at. And the reason why I say that is because sometimes the metanephros can be mixed up with the developing lung in pseudoglandular stage. The liver, I don't know if there are students that do this, but like, it could happen being mixed up with the heart if it's zoomed in enough. There are a lot of different mix-ups that a student can do, and knowing where you are will help you determine or help you cut down the options or limit your options of which organs they are. There are some additional structures I would like to add in here. I can't think of at the top of my head, but we'll add it in later in post. Okay, so yeah, that's what I wanted to say regarding how to identify where you're at in the body. Now we can actually get into the different organs for each area of the body. Now let's finally get to talk about the developing liver and its additional structures. Now the liver, like we said, is in the abdominal region of the embryo in Sagittal Cut. This is the Sagittal Cut. And we're going to get to the embryo in Transverse Cut, which is something we can see, for example, here, as well as here in a second, because usually we can also see the liver there as well. But let's first talk about this cut. So again, like I said, when it's zoomed in, it's hard to see where in the body you're located. But if we follow the ribs, in this case, there's like little bits of the sternum because it's so elongated, but this is definitely not the vertebral column. This is, right? We know that this area would be the thoracical region. So as soon as we get to the end of the ribs, that's where the abdominal area starts. And usually the largest organ, now this depends on the cut of the embryo as well. Sometimes we only get a little piece of the liver and not the entire thing. But in this case, we get a very large piece of the liver, usually the largest organ in the embryo. And that would be obviously the developing liver. Now the developing liver has some additional structures that I'd like to mention. But first of all, let's talk about where else we can see it. So here's another embryo in Sagittal Cut. Like I said, we follow the ribs. That's where the thoracical area is. As soon as the ribs end, that's where we have the developing liver. Only here it's a little bit more scattered. And then as soon as we get to the liver, they usually zoom in and show you additional structures of the liver. I think it's rare that they'll ask you about the organ itself. And instead, I think that they'll ask you about I think there's 2 or 3 structures within the liver that's specific to the liver. Here's another one. We can see the vertebral column here. Here we can see the sternum, so no ribs. But I think it should be obvious that this side would be the sternum side and this would be the developing vertebra. So as soon as we end the sternum, that's where the abdomen starts. And here we can find the liver. This is a little beak is also the liver, I would say. Okay, let's talk about the liver in transverse section before we get into some structures within the liver. Here we can actually see that the largest organ, again, is the liver, which it usually is. And like I said, this would be the sternum and these would be the developing ribs. You might ask, well, what are the ribs doing within the abdominal region? Could be because due to the cut of the embryo, also could be due to the fact that the liver pokes out a little bit. It all depends. But yes, here we can see the thoracical area with the 2 lungs. Now, when we have this transverse cut, usually the only things you can see is the liver and the lungs. So that would be an easy differentiator between the abdominal area and the thoracic area compared to a sagittal cut where you see a lot more organs and it could be a bigger mix-up. Also here you can more visibly see the developing diaphragm, which is made up of all of these skeletal striated muscle fibers. Between two lungs, we have the developing esophagus. Hope that makes sense. It's not that big of a star in terms of the structures that we actually have to know for this colloquium, but it might appear. I'm not gonna lie. Then we have what we brought up before, which would be the developing vertebral column with the developing spinal cord, the neural canal, and the 3 layers that we talked about before, including the pia mater, right? So that's what you see here. And then you see the lungs with the esophagus, and then you will see the liver. And again, here they will probably zoom in and show you the structures within the liver that are actually important. This is another slice, transverse section of the embryo. This is most probably an animal because we see a lot of hair follicles here. So we can see also the epididymis, which is this top layer, the didymis, which is this lighter area with all the hair follicles, and the entire skin development being both of them, right? But looking into the organs instead, like we said, here we have the developing spinal cord within a developing vertebral column. We have the 2 lungs here, and then also in the center we have the developing esophagus. Here instead though we don't have the liver, and that's because it's cut more towards the upper part of the embryo, so towards the thorax. Let's say we have a little embryo here with nubs and feet. So we have the belly here with the liver and the thorax here with the heart and lungs. So instead of it being cut here, it's cut here. Here up here instead, so we don't really get any visible liver fragments. We just get the top of the thorax. So that's why we see another organ here that isn't the liver, and that is the developing heart with the pericardium, I believe. And I think these are some chambers of the heart, but you can't really distinguish which ones they are. And here instead we might actually talk about the structures within the lungs instead of the structures within the liver, but we'll get to the lungs in a second. I think those are all for the liver once it's zoomed out. Here's another one. This one's less likely to pop up in the colloquium, but just in case. Here we have like a weird transverse section of the embryo. But we can see this characteristic shape of the liver. However, it will be a lot more zoomed in, so you won't really see the shape of it anyways. The diaphragm here, and usually this is where we would see the lungs, but again, it's a different cut, so we don't get the liver, we don't get the lungs, but we do see the developing ribs here, and then in between the developing ribs, we can see all of these striations. This muscle tissue, this is of course known as the intercostal muscles, but I think you would gain points if you just write in striated skeletal muscle fibers. I think that would be all right. Maybe you'll get all the points if you write in that, but I would have gone the safe route and just written the skeletal striated muscle fibers. Hi, and then here we can see what the transverse section looks in a microscope, or how the transverse section looks. And these would be developing vertebral or a developing vertebral column. And then we have the developing spinal cord. Then these would be the lungs. And then continuing down, we would see this large structure. This is the diaphragm with all the striated skeletal muscle fibers. And then the liver, developing liver, we have a blood vessel here as well. And then they might zoom in and show you something like this, right? With all of the structures within. Then here we also have the developing skin as well. I haven't added the annotations here yet, but I will do it later. Let's now talk about the structures within the liver. And we have them here actually. So here we have two different organs. We have the liver here and the developing pancreas here. I'll tell you how you will see the difference between them. I'll also tell you where we can find the pancreas. I haven't really talked about it yet, but let's focus on the liver first. Now the developing liver is made up of usually these large cells with a larger nucleus, lighter than most other cells within the liver, which are known as the hepatocytes. One of the most common cells we have to know in this program is the hepatocyte, and I hope it's memorable enough for you to actually know the names when you see them. But again, they're these large cells with a large nucleus and a pinkish cytoplasm, very distinguishable from all of these darker cells of the hematopoietic islands, but we'll get to that in a second. So differentiating hepatocyte is what you'll have to write. Differentiating as the adjective, I think, is important in order to gain all the points. Now for the remaining structures within the liver, or the specific structure of the liver, it is the hematopoietic islands. And these are these dark cells clustered together like so. So this would be a hematopoietic island. This would be as well, maybe this as well, this too. So they'll usually show you a bunch of these dark cells clumped together like this, and they'll like go back and forth with the mouse like this and then ask you about this structure. However, they may ask you about basic structures of the liver as well. They can do this for any organ too, so this is not just exclusive to the liver. For example, they can ask you about differentiating blood vessels. They can ask you about nerve, like a nerve fiber bundle, if they'll find that. I think that it's unlikely, but they might or may just ask you about it anyways, just to test your knowledge on the most basics of structures. The last structure in the developing liver that I'm gonna talk about is the megakaryocyte. And it's a specific multinucleated cell that we can find within the developing liver. Can we find it here? Yeah, kind of. So this is a microscopic image of the developing liver. You can see here all of these bunch of dark nucleated cells are hematopoietic islands. And then we have these very, very pinkish cytoplasm, almost neon, that have several nuclei inside them, known as the megakaryocyte. Also, we didn't talk about what the cells, or these dark cells, are called. They're called nucleated erythroblasts due to the fact that they're in the fetus. Do check that up with your professor, though. They could change the name of those cells, but the megakaryocytes will remain as it is, hematopoietic islands as well as the differentiating hepatocytes. Here we can actually see the polygonal shape of the differentiating hepatocytes. Well, actually we can't. I'm trying to find a good example, but it's very, here we go, maybe this one. So polygonal means that it has several corners, maybe several corners like this. So we can kind of see that this cell has these sharp several corners, and with a big nucleus and pink light cytoplasm that's very characteristic of the differentiating hepatocyte. All right. I would say this is also a megakaryocyte because you can see the cytoplasm. So it's not two cells separate. It's one and the same cell with several nuclei. That would probably be a megakaryocyte. Here's another example of the liver. This is a lot more fragmented. So again, it might not look like this. It might actually instead look like this. As long as you know that you're in the abdomen and it's a fairly large organ or structure, even though it's scattered, 9 times out of 10, you'll know that you're in the liver. And then if they ask you about a cell that looks like this, that has several nuclei within its body, then you'll know it's a megakaryocyte and that we're in the liver. It's a large clump. It's one cell with several nuclei. These would be maybe the hematopoietic islands in this case. The differentiating hepatocyte, I'd say, is very hard to see here. I'm trying to visualize it as good as I can, but it's, again, not as easy as it seems. These are better examples. So this is more zoomed out version of the developing liver. Here we have the blood vessel, which might be something that they ask you. This is also a blood vessel. All of these pinker areas are blood vessels. So again, they might ask you about it to throw you off, but it's a fairly, basic structure that you should know about. So hopefully you'll not have issues with that. But once zoomed in, we can see this. Now again, this is a blood vessel. I don't know if the professor wants you to be specific and say differentiating blood vessel or something like that. I think in the liver there are some specific or a specific name for the blood vessel. But for now, just I would say it's enough to just write blood vessel. Again, though, ask your professors. But yeah, we have the nucleated erythroblasts, which are all of these darker cells. And then we have the hematopoietic islands, which would be a clump of those darker cells. And here we can actually see the more proper shape of the differentiating hepatocyte. I wouldn't even say that this is a differentiating hepatocyte. It might be, but the nucleus just seems very, very dark for it. Instead, I would be more confident in naming these as hepatocytes. This could also be one, but like I said, it's not as obvious. Here we have another one. They're very polygonal in shape, like I said. Maybe you can see it here. This little cell, like I said, very polygonal. Here's another one. So yeah, definitely differentiating hepatocytes. Here, I would say that would be a nucleated erythroblast, judging by the color. Here's another one, the blood vessel with all the nucleated erythroblasts. And the reason why these nucleated erythroblasts appear more pinker and larger than these nucleated erythroblasts is because these are more immature in comparison with these, which have migrated into a blood vessel like so. Okay, and here we can see the differentiated hepatocyte. We can see one here, one there. These are as well as the hematopoietic islands. We can't really see a megakaryocyte here. I'm trying to see if I can spot one. Nope. Can we see it in any other slides? No, we cannot. Maybe we do see it here though. Here we do. Yeah, perfect. This is a liver in high magnification, and it's very fragmented, so we can barely distinguish the hepatocytes like so, and these would be the hematopoietic islands. And then we have the megakaryocytes, which would be this. So we have 3 nuclei here, apparently. This would be another megakaryocyte. Like I said, they appear very large and pink. So if you see a large and pink and multinucleated cell that looks like it's just a clump of other cells, you're within the liver, and it's most likely a megakaryocyte. This would be another one, I believe. Moving on from the liver, which is a fairly large structure within the embryo, I would like to talk about an organ that is adjacent to the liver usually, and that is the developing pancreas. Now the pancreas, you can find it underneath the liver, and it appears like so. It's a little bit darker than the liver. I can only find it in this slide. These would be a zoomed in version of the liver and the pancreas, but I can't really find. Oh, it should be here. This is a weird cut. It is still sagittal though. Here we have the developing pancreas. This looks very odd for the developing pancreas, so I wouldn't have studied the pancreas just by this slide. Let's see if I can actually find another one. There is another image here. If we just erase this for a second, we will be able to see the developing pancreas here as well. So this would be the liver, and then underneath this would be a little intestinal loop, and then we have this area of tissue that is a little bit redder and darker than the liver, and that would be the developing pancreas. Trying to see if we can find it in a demo lab image, and this would be a very odd version of it. Let's see here. So we can see it there as well as there, but again, it doesn't have a lot of context. This is the liver, so underneath the liver we have the developing pancreas, and we have these acini, which we will see in a larger magnification in a second. But here we go. This is actually a lot better. So like I said, the developing liver, and then we have the developing pancreas underneath, usually red in color and close to the intestinal loops, but directly underneath the liver. Now zooming in here, we can see the liver and the pancreas like so. And then within the pancreas, we actually have the developing acini, which if we zoom in, looks something like this. So they look like these flower-shaped structures, like so, with all of these petals. These are known, or this entire structure is known as the developing acini. Of course, you need to know about the tissue that kind of holds everything together, almost like loose irregular connective tissue, which you can pretty much find anywhere in the embryo, which is the mesenchyme. And then maybe you'll see some blood vessels here as well, but I think those are all, or that's all for the developing pancreas. Let's finally talk about the developing lung in pseudoglandular stage, and yes, you have to write down this entire name to gain all the points for this slide. The lung you will find within the thorax of the embryo, unless it's separated in a slide like this. You'll find it again in the thorax, which is lined by the ribs. So as soon as the ribs end, that's when the thorax ends as well. So that is not for you not to confuse it with the metanephros, right? Because we can find it within the thorax only. And we can also find it in a transverse section like so. So these would be the lungs. I'm highly doubtful you'll find metanephros or like adrenal glands within the transverse section of an embryo, because usually they want to like capture more organs or more important organs like this. So that's why just assume that if you're within a transverse section and you're close to the vertebral column, it's probably the lungs, especially if you see this developing esophagus in the middle of them. Here you will find it as well. Now, enough about the transverse section. Let's actually talk about the structures that are within. Now, first of all, we're going to talk about some common structures, and then we're going to talk about some additional structures that Professor Vivats has added. So the structures that are common for all professors are these. So for example, we have the blood vessels. However, Professor Vivats did mention that you have to know the difference between a vein and artery. Artery have larger blood vessel walls, while the veins have a smaller but a larger lumen. For example, here, I believe, if we can find a difference, this would be a vein and this would be an artery. See how thinner the dark walls of this blood vessel is compared to this? See how thick that wall is? The lumen here doesn't really matter. In this case, the vein has a smaller lumen than the artery, but it could just be the cut. It's not like the umbilical cord. But I would just go with the walls of the blood vessels instead. Surrounding the blood vessels, or surrounding all of these openings within the lungs, is the tissue we all know and love, which is the mesenchyme. So all of this lighter tissue within the lung, that's all mesenchyme. If they show you that, this would also be the mesenchyme that surrounds the blood vessels, this as well. This as well, like all of that tissue, just known as the mesenchyme. Now, what about these openings that aren't blood vessels? So not these, but these. How do we see the difference between them and the blood vessels? Well, in the lungs, the blood vessels are more apparent to us because they're always surrounded by the mesenchyme, like so. So very thick layer of mesenchyme. If we erase this, I'll show you. So you can see this is the vein and this is the artery. You can see the thick layer of mesenchyme that surrounds both of these structures. While here, this is a bronchi, and you don't really have that thick of a layer of a mesenchyme Obviously, also when you zoom in, these are lined by endothelial cells. Okay, now let's thank our professors, because during this course, we don't actually have to name the specific type of bronchi that we are looking at. So for example, this being a cartilaginous type bronchi, we don't have to write in the cartilaginous type. It's enough to just write that it's a bronchi. Same with the muscle type. It's enough to just write bronchi. However, there is a difference between the professors in regards to what are bronchi and what are bronchioles. So according to Professor Vaivads, the bronchi are these larger openings like so, while the bronchioles are the smaller openings. Professor Rona, however, accepts both as bronchi. So even the smaller ones as well as the larger ones are all within the same bronchi name. It's just Professor Vaivads that adds in the bronchiole for the smaller ones. Hope that makes sense. However, there is a specific type of opening of the bronchi that we do have to name besides the developing bronchi and the developing bronchioles, right? And that is the dichotomous division of the bronchial tree. I would say it's enough to write in the dichotomous division. Some say you have to write in the whole thing, but I think it's obvious that it is of the bronchial tree. Anyways, the dichotomous division. What is the best way for me to describe the shape of the dichotomous division? Now, don't laugh. I think it looks like a fidget spinner. So see this fidget spinner with its 3 little arms? It almost looks like a T, like so, right? That's sort of just how the dichotomous division looks like in my mind. So see all of these arms? Kind of looks like a fidget spinner, doesn't it? And that's why, or that's how I thought, how I figured out the shape of the dichotomous division. Now, this could be a dichotomous division as well, like this T shape. This too. So that characteristic fidget spinner shape is the dichotomous division of the bronchial tree. It's like the name suggests, it's just like a little division, like a crossroad that occurs where one little bronchi is split and separated into 2. So something like this, right? And that's what we see here. So that name is universal for all professors, and the shape of it is also very characteristic. Okay, you don't also have to write in the terminal bronchus. I'll probably get rid of those names. But like I said, these enough to just write bronchi for Professor Rone. I think they are bronchioles according to Professor Vaivads because of the size. This would be more of a bronchi according to Professor Vaivads due to the sheer size. And this, I would say, is probably a differentiating artery just by the thickness of the wall. It could be a vein, but the thickness just matches more with this blood vessel rather than this blood vessel. The very last structure of the developing lung in pseudoglandular stage I'd like to mention is one brought up by Professor Vaivads, which is the pleura development. Now, hopefully you remember the pleura from colloquium 3 of physiology, but it is this sort of tissue that surrounds the lung like so. It's almost like the pia mater of the lung. Is best how I would describe it. So this would be the pleura development as well. So this little loose shawl that surrounds the lung. This would maybe also be the pleura development. Okay, let's look at other slides of the lung and hopefully we'll get to the same conclusion. So for example, here we can see the pleura development. We can see all of the bronchioles or bronchi, depending on which professor you have. We can see the dichotomous division here, but also here as well, larger. The differentiating blood vessels that are here. This is apparently a vein. Good to know. Then the developing bronchi as well. Is there anything? Yeah, the mesenchyme here too. And this would also be the developing pleura. Okay. Here we have the same thing again. So let's try to see. Here we go. These are the structures more in a zoomed in manner. So here you'll actually see the pseudostratified epithelium. I don't think this is a structure that you actually have to know, but you can see it lining this bronchi. You can also see the differentiating blood vessels. In this case, this would be a vein because of the very, very thin blood vessel wall. We can only see the endothelial cells here practically, and then also the mesenchyme surrounding it. And like I said, in the transverse section, they might zoom in here and show you the structures of the lung from a transverse slide as well. But if we look at the demo lab slides instead, these are some examples of the lung. These are separated lung tissue, so not in a total embryo slide. And then we also have the structures within, which we can see here. This is cartilaginous type because the professor mentioned that it was, but you don't again have to write in which type of bronchi it is. Here are some more images under the microscope. You can see the pleura development here more clearly because this is the corner of a lung in pseudoglandular stage, and we can see the differentiating blood vessels here as well with the characteristic red blood cells within. This would be a dichotomous division. This would be a bronchiole or a bronchi, depending on the professor. We also have a zoomed in version here where we can see the epithelium more zoomed in. Here it actually appears almost cuboidal, and as well as the pleura and the mesenchyme within. You've all been absolute champs. Now we get into our last organ of the class, which is the small intestine development. Now we will find it within the abdomen of an embryo in sagittal cut, or we'll find it separated in a slide like so. Now there are quite a few small intestine structure that we have to know, but some that we should be familiar with. First of all, the developing intestinal villi, which are all of these finger-like structures that sort of line the small intestine like so. Here they are a little bit cut oddly, but if they were to ask you about it, I think that they will show you one that is actually connected to the small intestine like this. Then there are the cells which line the developing intestinal villi, and let's zoom in to one. Here we go. This is what we just saw, the developing intestinal villi. They are just cut very differently, so they're cut like this instead. And there are two types of cells that line it. The first one is the enterocytes, which are the most abundant cells. There are these simple columnar cells that helps with the food uptake. We can see it here. And then a row of those cells is simply just called the simple columnar epithelium. Then we have the cells that contain a lot of lipids in them, and those are known as glandulocyte if you ask Professor Ronne, but if you ask Professor Vaivads, they're actually called goblet cells, which are the more developed glandulocyte. So it all depends on the professor, but they're pretty much the same cells. So these are glandulocytes / goblet cells. All of those would be glandulocytes. If you're feeling unsure about it during the colloquium, you can just ask the professor to show you another example of it. And if you see that it's quite a rare cell, like it only appears in a few per villi, then you'll pretty much know for sure that it's a glandulocyte you're looking at and not an enterocyte. Boop. Okay. So the next structure are the layers of the small intestine. Now there are more layers than this, but the first one is the submucosa that is just underneath the mucus part. So I would say maybe this even would be the submucosa. More importantly though is the muscularis externa, which consists of this longitudinal and circular layer. Maybe this is circular and this is longitudinal actually. It's just 2 layers of muscle tissue or smooth muscle tissue that's underneath the submucosa. Quite an important structure to know for this colloquium. The other important structure is the tunica serosa, which is directly underneath the muscularis externa, and you can kind of see it as this loose tissue, again, underneath the muscle tissue. So there we go. All of that is the tunica serosa. Thankfully, you don't have to actually know which section of the muscularis externa you're at. It's enough to just know that both of them make up the muscularis externa, and the thing underneath would be the tunica serosa, and the thing above would be the submucosa. Here I wrote, the inner circular layer is this, and the outer circular layer is that. Okay, so none of them are the longitudinal layer. Apologies for that. The very last structure within the small intestine is the apoptotic epithelium, or according to Professor Vivads, the epithelial remnants after recanalization. Now, this is a very important structure because a lot of people mistake the apoptotic epithelium for these structures, even though they appear almost exactly the same. Now, these structures have these lamina propria centers within them. So, for example, if we were to cut the tip of this villi, we have that lamina propria in the middle, and then we have the cells that line the lamina propria all around it, like so. So, like a flower, right? While the apoptotic epithelium is just white cells with no lamina propria. There's no center in it. It's just uniform whiteness. I'll show you guys another example. This would be a apoptotic epithelium, or according to Professor Viva's, epithelial remnant after recanalization. Like you see, it's a lot whiter. There are some nuclei within them, but there is no center of lamina propria like there is here, for example. Here we can see the lamina propria center. This is another apoptotic epithelium as well. Let's see if we can find it here. I doubt it, not in that slide. Here we do. Okay, so this is the developing small intestine, and then once zoomed in, we can see this. Now you can see here that here we have the lamina propria within it, right? And here we can see the lamina propria as well, lined by all of that simple columnar epithelial cell, almost looking like a flower. While here, this just looks like a bunch of cells, almost like bubble wrap, clustered together like so. Very light. Sometimes you can see nuclei, but in general it's very light. That is the apoptotic epithelium. I believe that's it for the small intestine development. Let's talk about some other organs or structures that are not as important but might still appear. Personally, I hate this slide. I don't really recognize the umbilical cord here at all. I don't recognize the midgut. Saying that, the professors might just pick this up for your colloquium and show it to you regardless. So again, take this with a grain of salt. I don't think this slide is that important. But let's talk about what other structures might still be in regards to the embryo and sagittal cut. Now here we see, like I said, the liver in the abdomen. We can also see the developing pancreas here, and we see the small intestine development. Now there are other parts of the intestines that we will also see within the abdomen. For example, here we can see the recanalization of intestine. What does that mean? That means that it doesn't have a clear lumen yet. The intestines are in their recanalization phase. There's a lot going on within the lumen of these intestines. Otherwise, if the lumens were clear, like it kind of is here, then this would be more of a loop, or an intestinal loop. Here we can see the developing umbilical cord. Isn't that cool? So we can see the artery here. We can see the vein. I think that would be also an artery. If it was cut in a transverse section, it would look very much like the umbilical cord that we saw in colloquium 1. With the Wharton's jelly and everything. And this might still appear in colloquium 2 from professors who want to just test you from, the colloquium 2 knowledge if it's a structure that does appear in their slides, but it's not that important, I would say. I wouldn't have studied on it personally. Here we have, I believe, a developing external genitalia of the embryo that also might be something that appears. Here we have more recanalization of the intestines because there's a lot going on within the lumen. Here's more recanalization of intestines. This could be the stomach of the embryo. That could be another structure that appears during the colloquium. The hindgut could also appear in the colloquium, although I'm not really that personally confident in the hindgut, so please ask your professors in case that is a structure that they want to talk about. But other than that, I think we're good. Here we go. This is the hindgut. We did talk a little bit about it in colloquium 1. Kind of looks like a dorsal aorta, just more towards the bottom of the embryo. But again, it's kind of hard to recognize and distinguish it from the dorsal aorta. That's it. That's all for this class, thankfully. Now we're gonna go through class 6 after this, and yeah, then you guys are done, hopefully, with your studies, and we can get into the test yourself video.